Welcome to the Japan Society's regular webinar on current affairs, business, and many related matters. I am Bill Emmott, and as many of you know, I have the honor and pleasure of chairing the Japan Society. Often, as I say, our topics are about issues of politics, international relations, business, and the economy that happen to be in the news. And often in relation to these, we and our speakers talk in a general sense about the value of and potential for collaboration between the UK and Japan. This week, I'm delighted that we are covering a topic that is not in the news, but undoubtedly should be. And it is a topic that, as we shall see, has given rise to a great deal of valuable and practical collaboration between our two countries, with the potential for far more. This issue of medical collaboration is in a great tradition of scientific collaboration between the UK and Japan, especially in the medical field. Collaboration that, as I noted in a recent blog, has long been both marked and encouraged by the Butterfield Awards in Medicine and Health, established by the Great Britain Sasakawa Foundation in 1985 in memory of Lord Butterfield, himself a professor of medicine at Guy's Hospital in London, who had close connections with Japan. There will be more details about those awards for anyone interested in the chat function, including the deadline for applications for 2021. But today, we are welcoming three speakers, two in the UK and one in Japan, with a discussion that marks both collaboration between the UK and Japan and the establishment of a new entity at Oxford University, the Institute of Developmental and Regenerative Medicine. Our first speaker, Professor Paul Reilly, the director of that institute, will explain far better than I can the kind of path-breaking work the institute plans to do. Our second speaker will also be from Oxford, his colleague, Professor Georg Holländer, head of pediatrics and professor of developmental medicine. And our Oxonians will be followed by our third speaker, Professor Shin'ichi Takeda, honorary director general of the National Institute of Neuroscience in Tokyo, which has a long history of collaboration with those Oxford departments. After our speakers' opening presentations, we will have a discussion and, crucially, your questions. Please, as usual, submit those questions using the Q&A function, and feel free to vote for other people's questions so as to bring them to my attention more quickly. Welcome to all three of you to the Japan Society. I pass over to you, Professor Paul Reilly, to kick off the presentation.
Okay, thank you very much, Bill. And I'd like to thank the Japan Society for the opportunity to tell you about some of the activities we have ongoing, both at the University of Oxford in regenerative medicine, and also linking across to our colleagues in Japan. I'm very pleased that Professor Takeda is joining us from Tokyo in the evening over there to elaborate on an ongoing collaborative project with one of the key members of the new institute. So if I could have the first slide, please, Lucy. Okay, well, that's me. Right, so I just wanted to highlight really the sort of general definition of regenerative medicine as we see it. And who better to quote than Sidney Brenner, 2002 Nobel laureate, and also alumnus of Exeter College. Regenerative medicine is the process of replacing or regenerating human cells, tissues, or organs to restore or establish normal function. And I think it's fair to say that regenerative medicine is extremely challenging, and there have been very few clinical successes in this space, which means that it's very important to think about alternative ways to tackle potential regenerative diseases, etc. So if we can move to the next slide, please. But of course, an enormous breakthrough in this space occurred in Japan back in, well, over nearly 10, 15 years ago now, with the finding by Professor Yamanaka that you could reprogram cells from the human body to become stem cells using the so-called Yamanaka factors, and that these stem cells could then be directly differentiated into different cell types of the body. And of course, this has huge implications for drug screening, for toxicology screening of existing drugs, and for the use of cell types in cell therapy for repairing damaged and diseased tissues. And you'll hear some of the uses of these cell types, particularly in deriving skeletal muscle from Professor Takeda shortly. But we use the iPS cell model derived by Professor Yamanaka in our current studies, and we plan to embed these and this model system in the new institute, which brings me to the next slide. So we have an ongoing project that is a strategic priority within the Medical Sciences Division at the University of Oxford, led by myself and Professor Georg Holländer and also Professor Matthew Wood. And Professor Matthew Wood is not with us on the call today, but he is a professor of neuroscience working very closely with Professor Takeda. And the Institute of Developmental and Regenerative Medicine is an ongoing construction project and will be opening its doors in January next year. It is at its core a merger of developmental biology and regenerative medicine, and our philosophy is very simple. We seek to understand how tissues and organs are formed in the first place during development, during pregnancy, to then inform strategies to repair and replace the same tissues and organs when damaged or injured through disease. And so it is a 3-floor building which will have themes in immunology, cardiovascular, and neurosciences, integrating across those. 50% capacity will be from incoming Oxford researchers, but also it represents a very valuable recruitment tool. And of course, a tool in which we hope to establish international links and further collaborations, particularly with our colleagues in Japan. So if I can take the next slide, please. So the challenge here is that 2/3 of the deaths worldwide are due to non-communicable diseases. Obviously, we're very conscious of the current pandemic and the communicable disease that is COVID and coronavirus, but many deaths are caused by non-viral type diseases. And a lot of these are cardiovascular, neurological, or immune system disorders that have a developmental origin. Hence, there is an urgent need for new drugs and therapeutic strategies to replace and restore damaged tissue arising from birth defects or acquired adult tissue injury and disease. And of course, one area that we're able to impact on here by studying developmental biology and normal development of these organ systems is that we can look to make a therapeutic impact on congenital disease as well as on regenerative medicine in the adult setting. Next slide, please. So the goals of the Institute are to improve the current understanding of the molecular and cellular inputs that control normal development of the heart, brain, and immune system, to decipher mechanisms that cause congenital disease, birth defects, and an increased susceptibility for acquired adult disease, to identify developmental embryonic gene programs and cell potential within the developing embryo as targets for us to reactivate in adult heart, brain, and immune tissues following disease or injury. So many cell types that exist during development are often dormant in the adult and are required to be reawoken for a process in which we look to repair tissues from within by stimulating those cell types. And ultimately, we want to establish novel therapeutic strategies designed to regenerate, repair, and replace affected tissues. Next slide, please. And as I say, the Institute will embed themes in neurosciences, cardiovascular sciences, and immunology. And here, it's important to note that there will be similarities in these across these 3 systems, which we can exploit, but there will also be key differences. Obviously, all 3, the immune system, the central nervous system, brain, and the cardiovascular system are highly specialized, but there will be common approaches that we can use. And as I say, the program in neuroscience will be led by Professor Matthew Wood, and you will hear more about the immunology component from Professor Georg Holländer on this call, and I will be leading the cardiovascular component. And the idea here is to integrate programs, world-leading research programs that will lead to therapeutic endpoints in treating human patients. Next slide, please. Of course, none of this can be done locally and without outward-facing international collaboration. And we are extremely proud of the fact that we've got significant collaborations with Japanese colleagues, and we'd like to establish those much further, particularly thinking about the exchange of trainees, so students and postdocs, and establishing fellowship programs to enable those trainees to experience the best of what we have to offer in Oxford and elsewhere in host Japanese institutions. And I think that people are the main driver of this process. So we're very keen to reach out and establish collaborations. And of course, because we are recruiting into the Institute, very keen to hear from interested Japanese scientists in this regard. Next slide, please. And just to illustrate the importance of this type of collaborative approach, in my own field of cardiovascular development and regenerative medicine, the preeminent meeting is the Weinstein International Meeting. And in 2018, this was hosted in Nara, in Japan, by two colleagues, Isao Shiraishi and Hiro Yamagishi, both of whom have become good friends, and we've continued to work together. The two of them hosted this meeting very successfully in this wonderful venue of the No Theatre Stage. And of course, we had access to the very world-famous Sika deer in the grounds. And it was a wonderful meeting which established a huge number of ongoing collaborations, and both are now international members of the Weinstein Committee for future meetings. Next slide, please. And in the same meeting, I managed to catch up with an old friend of mine, Professor Kenta Yashiro, who I got to know very well during his time at Queen Mary University of London up until 2016. And Kenta's work on early heart development and establishing how cardiac progenitor and stem cells are specified and then can differentiate to form the tissues of the heart is crucial to the type of work we're proposing around understanding heart development and how that extrapolates to regenerative medicine. And Kenta is currently at Kyoto University of Medicine. And this is the two of us as he was showing me around Osaka in a wonderful trip back in 2018. Next slide, please. Okay, so I have finished for now, and I'll just hand you over to Professor Georg Holländer.
Yes, good morning, good afternoon, wherever you are. I thought I would give you a link to the immunology program in IDRM, and where we think that immunology will play a very key role in the overall aims and goals of the Institute. Now, as you know, the immune system's capacity is the ability to discriminate between self and non-self. Next. And this means that in one instance, the immune system, when recognizing self, next, should not respond or be tolerant, while in the other instance, where it recognizes non-self, it should create a directed and effective immune response. Next slide. Now, in molecular terms, self is rather complex. Next slide. It is namely the combination of all the genetic information in our cell nuclei, which composes 23 different chromosome pairs and encodes for approximately 22,000 proteins. Now, these genes collectively, next slide, are responsible that our body has over 4,000 different cell types. And we learn almost every month about a new cell type in our body, which has, as shown here at the bottom, an enormous number in the adult to be coordinated. Now, each cell, next slide, is composed itself again of different compartments. And in each of these compartments, we have up to 1 billion proteins. So all of this is self, and all of this needs to be recognized by the immune system as such and not respond to. While, next slide, the non-self component, as you can imagine, is equally complex. So as non-self, from an immunological point of view, next slide, we consider all other animal species against which we can respond. So for example, if you get bitten by a dog, you will respond to the dog's proteins in the saliva. It contains the millions of different viruses, and we are experiencing at the moment a particular challenge from coronaviruses, or the up to 1 billion different bacteria. And all these will need to be recognized by the immune system and fought against. Next slide. And so what the immune system does is similar to what our cognitive system does. It recognizes pattern and establishes them as normal, such as the picture here of Mona Lisa. And if non-self is presented, next slide, our brain recognizes quickly that the Mona Lisa has been slightly changed, and we do respond to this as saying this is not the correct picture. And that is exactly what the immune system does with two different effector mechanisms. Next slide. On one hand, we have the innate immune system, which responds very, very quickly, but can only recognize a number of different foreign antigens or non-self antigens. And on the other hand, we have the adaptive immune system, which takes a couple of days to respond, but has an unlimited capacity to recognize non-self, but also actively prevents its activation when recognizing self. This results in a memory, an immunological memory, which works particularly well if you think of vaccines. We are vaccinated against an antigen, and years later, we still can respond to that antigen in a specific way. The flip side of the coin is, however, that autoimmunity can be driven by the adaptive immune system. At the center, next slide, of the adaptive immune system are T cells. And these cells, next slide, are generated in the thymus, which is an organ sitting on top of the heart in the chest and produces throughout life normal, new, naive T cells that orchestrate the adaptive immune response. However, next slide, this organ decreases in size and function with age. And so when you are older than in your mid-teens, namely after puberty, the thymus output decreases dramatically and is then only a trickle at ages beyond 50. This is important, next slide, because we only have a limited number of T cells in our body and only a relative limited number of their receptor specificities to recognize all different antigens. And we need to keep that pool of T cells alive because, next slide, with age, we decrease our vaccine responses. We are more impaired in our responses against infectious diseases or malignant cells in the context of cancer, and the chances of developing autoimmunity increases. And so it's really the focus of the work in the immunology branch of IDRM, next slide, to try to better understand how the thymus can be regenerated. So how these blue cells, which are the T cells, are educated to distinguish between self and non-self. And this education occurs by these green cells, which are thymic epithelial cells, which form kind of a scaffold that interacts with the developing blue cells. And this is an effort that is not only to be exercised in IDRM, but, next slide, is actually a global activity with a lot of colleagues and friends in Japan who contribute to our growing understanding of thymic epithelial cells, how their stem cells look like, how we can mature them, manipulate them, how they grow, and last but not least, how well their function is to teach the immune system to distinguish between self and non-self. And these collaborations are with the university in Kyoto, Sendai, Tokushima, and Tokyo. As Paul explained, next slide, this is also an activity that we have been able to undertake with the building of IDRM in collaboration with Dr. Tetsuya Nakamura, who is the CEO of the Itabashi Medical System and has allowed us to create the institute as such, but also is the person with whom we will establish direct fellowships of physician scientists to come to IDRM and develop further our knowledge in regenerative medicine. And so as a last slide, next, what we really try to achieve here with the three arms of IDRM is a process in regenerative medicine that starts from the concept that repair from within is the best solution to regenerate tissue. Next slide.
Okay, so dear everyone, first I would like to thank the Japan Society for sending me an invitation to join in this meeting. I'm Shin'ichi Takeda. As Bill explained, National Institute of Neuroscience in Tokyo. I have been participating in the progress of stem cell research and regenerative medicine for a long time, especially in the area of skeletal muscle and muscular dystrophy. Paul has already mentioned that the concept of iPS cells has been proposed and established by a brilliant Nobel Prize winner, Professor Shin'ichi Yamanaka. But we know the origin of this idea has been found in the muscle biology area. Around 20 years before the investment by Professor Yamanaka, Professor Harold Weinstein found myodi, which can convert different kinds of cells, including fibroblasts, into myogenic cells. This is the original idea of direct reprogramming. Next slide, please. Well, in skeletal muscle, there are two kinds of stem cells. The first one is satellite cells. Dr. Soichiro Fukada, one of my previous colleagues, now he's in Osaka University, isolated fresh satellite cells by using specific antibody and identified numbers of genes which specifically expressed in satellite cells. There is another stem cells in skeletal muscle that's mesenchymal progenitors. Another, my colleague, Dr. Kiyoshi Uezumi, found these cells. Now he is in Tokyo Metropolitan Institute of Gerontology. Next slide, please. So this is a graphical summary of these two cells, has been shown in this slide. Undoubtedly, the key player of muscle regeneration is satellite cells, but mesenchymal progenitors support muscle regeneration process together with macrophage. These mesenchymal progenitor cells also have distinct role in the step of fibrosis and fatty infiltrations in skeletal muscle as well. So next slide, please. The third progress of our research group is introduction of myogenic cells from human iPS cells. This slide shows some of our recent progress, but we noticed prostaglandin EP2 receptor inhibits differentiation process of muscle progenitors. We now accumulate the data that inhibition of EP2 signal pathway accelerates induction capacity of iPS cells into myogenic cells. Next slide, please. This slide showed our current understanding of regenerative medicine in muscle disease, including muscular dystrophy. We intended to have potentially mature myogenic cells in vitro, and we are now trying to apply these cells into transplantation into disease condition. But certainly, we need further progress by collaboration. So next slide, please. Well, as already Bill and Paul mentioned, we have actually a long history of the collaboration between our research group and Oxford University. One of our talented researchers, Dr. Yoshitsugu Aoki, stayed in Professor Matthew Wood's lab for 3 years as a postdoc after he has gotten a PhD in my lab. He now became the director of our laboratory 2 years ago as my successor. So we are now expecting the results of their current ongoing collaboration and also future extended collaboration program. So next slide, please. And this slide shows the future perspective of our collaboration program. The progress of stem cell research that has been shown at the inside of this circle. We need to effectively differentiate stem cells into myogenic cells. On the other hand, for regenerative medicine, which has been shown at the outside of the circle, but especially patient-derived cell should be genetically corrected since the causative gene has been mutated in those cells. We can expect the progress of gene editing approach in the near future. Here, we would like to push the progress of both stem cell research and regenerative medicine, but at the same time, gene therapy and regenerative medicine cell therapy could be connected to each other. My colleague, Dr. Yoshitsugu Aoki, is now particularly interested in 3D organoid culture of human iPS cell-derived cells. Therefore, gene editing and organoid culture would be very good targets of future collaboration between our National Institute and Oxford University and your institution. Finally, I would briefly mention the regulatory issue. The Japanese previous Abe cabinet in Japan made a considerable efforts to prepare new laws to facilitate regenerative medicine. And we have been concerned in this new law through Japanese Society of Regenerative Medicine. So next slide, please. This slide shows the beautiful landscape of Oxford University and the view of our national center. We are at the peripheral part of Tokyo, Kodaira. And the best season in Kodaira is either a spring with full of cherry blossoms or autumn with plenty of autumn leaves. So we really like to invite you to come to Japan and to visit us in Kodaira. So I should stop here.
Thank you, Shin'ichi, and to Georg. Wonderful presentation. So I just wanted to close by thanking Dr. Nakamura in particular for making the Institute and the building possible, and the British Heart Foundation for their support. And I think the link with Dr. Nakamura is something that we're very, very proud of, and we wish to work further with him and the IMS group in the future. And the challenge now, of course, is getting this institute fully operational, to which we require, obviously, funds and, importantly, people. And as I outlined before, we're very keen to hear from interested researchers and scientists in Japan through exchange programs and recruitment, and of course, the sort of trainee fellowship exchange that I mentioned and also Georg mentioned. Particularly around clinical scientists who have an interest in developing therapies. And you can see shortly our own website will be launched, and we will be on Twitter and social media platforms. And please do feel free to email Lucy, who is our representative in the development office, who does a fantastic job for us in publicizing the Institute and seeking support for us to move the project forward. So very keen to hear feedback, and also very keen now to hear questions and any comments on what we've just presented. So thank you very much for your attention.
Well, thank you all very much, Paul, Georg, and Shin'ichi, for fascinating presentations and very inspiring to a layman like me, and I hope, I'm sure, to many of the more specialist people in our audience as to the potential, both for the work that you're doing, and for UK-Japan collaboration. We've got some questions coming in, and I encourage people to submit more, but I'm going to just kick off with a general question that situates your work and your topics really in the COVID pandemic that we've been living through. I wonder if each of you could comment on how, if at all, the experiences of the past 18 months that we've been going through have affected your thinking, your institute, your field. So perhaps we'll start with you, Paul, since you're on screen.
Thank you. Yeah. So I think, well, there's a few items just to highlight. Obviously, the lockdown period in which we were really unable to access the laboratories between March and end of June, July last year was a very difficult period for those individuals and trainees, particularly in the group who work in the laboratory environment, because they clearly couldn't operate and couldn't do any work. Those people on studentships and fellowships, which are fixed term, are obviously nervous about the impact on their work during this fixed term period. So I would flag our trainees as the people most potentially impacted by this, and something we need to be very aware of in thinking about how we can support them through extensions and further funding. And then I talk about funding, knowing that, for example, my main funders, the British Heart Foundation, have had a 50% cut to their research budget simply by being unable to fundraise in the normal way that they're able to do. And indeed, they've launched a national television advertising campaign to that effect, because the impact is huge. And then finally, just the ongoing process with the building. I think the contractors have been fantastic in accommodating distancing in terms of the construction, etc. We've only had a relatively short delay given all of that. But of course, there are delays, and COVID has impacted on that through factory closures where we're seeking raw materials for the building. And of course, the contractors have to operate under difficult distancing and conditions, etc. And then, of course, recruitment, encouraging people to move during a pandemic when you're not able to fly from overseas, from many countries, and to actually mix and to actually come and view Oxford and come and view the building is also challenging. So a whole host of things that have made it very difficult. But, people are resilient, and moving forwards, I think that, there will be an opportunity to bring people across and to view the Institute and to recruit, and also, I hope, to secure funds necessary for the research programs that are ongoing.
Thank you. And George, we're all amateur immunologists these days, so we assume that there'll be more issues in your field.
Absolutely. So with regards to the pandemic, one of the best systems to bring the pandemic to a close is obviously a functional immune system. So a lot of immunological questions have basically presented themselves as part of the pandemic, and their answers, we think, will clearly provide a way out of our present situation. So as example, we have learned that the antigen of the virus against which we create an immune response requires, for example, T cells. And we want to make sure now that these T cells are produced and available at the right number and with the right function. And so issues of how well do elderly respond to vaccines have come to the forefront. And we have seen that the vaccines that are presently approved, that they all seem to work very well, but we don't know yet how good the memory is over time, and how well the system can adapt to new evolving viruses. So here clearly immunology is challenged, but I think we are well positioned to provide very important answers to these central questions. So immunology has had, as you said, a bigger impact and a bigger importance. And I'm very glad that many people now think that they are immunologists.
We'll emphasize amateur, I think.
Well, amateur is in the eye of the beholder, so that's fine.
Very good. Shinichi, how do things look from Tokyo?
Yeah, probably I need to explain the overall situation in Japan in regard to coronavirus. As actually you may know, the frequency or numbers of the patients affected by coronavirus, it's a bit smaller when I compare the number with in Europe or in England. But instead, Japanese people very much afraid of the coronavirus. And actually our National Institute is in the National Center of neurology and psychiatry. So we need to find a way how to overcome the infection, not only in terms of infection or immune disease, but or immunological condition, but we need to somehow improve psychiatric condition, and then they can fight against coronavirus. But personally, last year from April to early June, we should stay in our home. And then middle of June, we started to come to the institute, but we need to keep that distance. And in my case, more than 90% of my time, I just stay in my home or in the office alone. So again, it was a big problem. But I hope now vaccine or distribution of vaccine in Ireland or in Japan may improve the situation of Japanese sensation or feeling a bit more. That's I really like to have those non-pandemic situation of coronavirus.
Right, so thank you. We've got a lot of questions, and one I think I immediately should pop in, which follows on a bit from what you, Paul, said about funding. Nicholas McLean, our Japan Society member, has asked about Brexit. Has Brexit adversely affected the developments at the IDRM by excluding Oxford from EU funding sources for scientific research and hampering free movement of EU scientists to the UK? The Brexit effect, can you sum that up, if there is one?
I guess part of the issue with Brexit is that because the pandemic landed around the time that we might have started seeing the effects of Brexit, it has sort of overshadowed that to some extent. So at the moment, there's no movement of people, quite literally. So in terms of free movement, scientists, I'm sure I'm speaking for the vast majority, would have been opposed to Brexit for obvious reasons. We had seek an open, collaborative environment, funding with our European colleagues and exchange with our European colleagues. Many of us, I'm sure I speak for Georg and Shin'ichi, have worked with European scientists in either consortia or in partnerships. And, so I would anticipate, because we haven't quite seen the effect yet, largely because it's been overshadowed by the pandemic, there will be very negative circumstances and issues related to Brexit. I think at the moment it's unclear what the precise nature of the deal between governments will be, which may enable us to continue to seek EU funding. I think at the moment we still can, which is very, very good news. But the freedom of movement and the kind of barrier that Brexit as a concept presents is also difficult for moving trainees. And for people in Europe thinking that the UK is welcoming in that regard, it's obviously taken some of that away in terms of perception. So I anticipate there will be some negative components of it, undoubtedly, but the pandemic has overshadowed that. So I think we've got to wait and see how that pans out. And I really hope government is sensible about enabling the funding situation to remain and for forging strong links with individual European countries rather than perhaps the EU as a whole, that we can also then establish partnerships, etc. So wait and see, I guess, is the short answer.
And thank you. Now, Koji Fusa has asked a very pertinent question about the IDRM. What's the difference between the IDRM and other research institutes focusing on regenerative medicine? You're all collaborating. Is there something that you would like to leave the audience with as to says, here's what's the special ingredient of the institute?
I can take that, or George, would you like to take that?
So you will find many institutes that are dedicated to developmental biology or regenerative medicine. But the combination of the two and how understanding how the organs are being built and then using this blueprint to be used therapeutically to regenerate tissues, this is something very unique. And this combination we have not seen in this form anywhere else, with probably one or two exceptions. And so it is, I think, this uniqueness of combinations understanding physiology and then using that knowledge to drive therapeutics that makes IDRM outstanding.
Thank you. So Paul Diamond, our member, has asked a specific question about the state of research in both Japan and the UK about spinal injury, about repairs, about work on the spinal injury. Might you like to address that question, Professor Takeda? What's the state of research in Japan on spinal injury?
So spinal injury is a very serious injury, both in UK and in Japan. And probably I'd mention our attempt in Kyoto University. So they tried to induce a spinal neuron from iPS cells. Originally, they had an idea to induce spinal neuron from the individual, from the patient himself. But it takes a long time to induce neuronal cells individually. So now they have resources like bank of iPS cells, and then they can pick up cells and then induce into spinal neurons and then transplant into diseased patients. At least their attempts in experimental animal models and like primate have been very much successful. So now they have an idea to do some clinical trials in Japan. So they have a very huge asset to do clinical trials. So we can listen their results relatively soon. That's our understanding.
Oh, yeah, that's quite encouraging. Paul, is there anything you'd like to add on this area?
No, I think that it's clearly a very important problem, and I know that there are colleagues in the Edinburgh Regenerative Medicine Center, for example, working in this area, similar to Shin'ichi's account, where they were trying to develop neurons in culture from iPS cells, and as he nicely described, that takes a long time. So there have been other approaches. I think part of the problem, though, just to highlight a general problem, is that with any type of injury, spinal cord included, there is often scarring and fibrosis. And this is a barrier to new tissue regeneration, including in the spinal cord. And, we have approaches. This is true in skeletal muscle. It's true in the cardiovascular system in the heart, to look at how we might modulate that scarring response so that we can establish an environment that's better for the new cells to recover and regenerate the tissue. And of course, very much related to scarring, and a driver of scarring is the immune system, which is why we've integrated, obviously, a strong immunology component.
Thank you. Shigeru Araki has asked a question that I find quite interesting and intriguing. He looks forward to the practical application of regenerative medicine. At the same time, he's interested in whether ethical regulations keep up with the progress of such medical technology. If not, what should be done as the first priority? How do you see this issue of the ethical aspects of your work? And or perhaps there are other parts of Oxford that will be paying attention to that. George, do you want to address that?
Yes, this is a very important question. Obviously, only because we can do things are we in a position that we should do them. And I think the first step to being able to do regenerative medicine is to understand how normal development occurs and where we would have derailment of the normal process that results in tissue damage. So all this is very much kind of standard medical understanding of health and disease. Now, the point that I think you're making here is what happens now if you start to alter such a trajectory, and what happens if you change, for example, the makeup of our cells and their potential to regenerate. And there we are, I think, in a very clear framework of regulatory rules that apply to biology, namely, do we create a effect with causing more damage over time? And is that a price that we as a society or as an individual can, should, or ought to pay? And here I think, again, my understanding is we need to understand the biological principles extremely well to start to understand what implications this might have. And regenerative medicine, as many other aspects in medicine, needs to be open to the scrutiny of ethical questions and try to find the right answers. We cannot operate in a sterile environment or in a vacuum, not exposing us to these societal questions and providing the correct answers.
Thank you. Shinichi, how do you think about this, this ethical dimension from the point of view?
Obviously, ethical issue is very important and very critical. And we know application of regenerative medicine has been extended in some countries, but we need to have a kind of rule. So first of all, enough progress of basic research will be necessary. And then clinical trial should be controlled and particularly done. And robust data would be necessary to get by clinical trial. But we have a little bit different station we have. Japanese government and foundations push the new law to facilitate regulatory issues, but like shortening of the process for decision, like decision making should be relatively short. But still, we really need to keep ethical issue. Otherwise, we cannot push the progress of regenerative medicine. That's our whole understanding.
Thank you. Now, quite a specific question, but an encouraging one from Kari Millard. Is there any opportunity for students to interact more with your institutions? I would love to be involved in the research as a Japanese student studying regenerative medicine at UCL. So how do you answer such a thing? That's Paul, maybe you should.
Perfect. Absolutely. 100% there's an opportunity. I mean, when we open our doors from January next year and we can then actually have people come and visit us and interact with us and see what facilities we have and experience in terms of an opening symposium, the sort of research that we're doing. So when we are able to put that out there, I'm quite sure that we'll have students and others interested in the work we're doing. And of course, collaboration with our colleagues in UCL and groups in UCL and beyond is very much something we'd like to encourage. So I would say watch this space regarding the launch and the opening and the inaugural symposium. And I'm hoping that together that will sufficiently publicize what we're doing, that there'll be lots of opportunities. And we very much welcome students who are keen to interact with us and with the member groups of the Institute and indeed incoming groups. So great question.
Thank you. Great. Now, Richard Siva has asked, thank you for the fascinating presentations. We'd like to ask about how well the process for scale up from laboratory discovery through small-scale drug research manufacturing into the fully approved process. Does this process for scaling up need to be modified in the UK or Japan? How do you think about that? Who would like to address that?
Well, maybe we'll do a double act. I can tell you a little bit in the UK. I would just state the case that Oxford, for example, and indeed many institutions in Japan have got a great track record of taking discovery science into spin-out companies and small biotech concerns so that you can move that forwards into sort of drug discovery. And even within the environment, the local environment, we have, for example, a target discovery institute which is built on the premises as a small pharma kind of setup around screening for new drugs and repurposing of existing drugs. So there is already that kind of, if you like, template for how we might do this in terms of moving from the discovery science through to the smaller scale. And there are even funding that's driven specifically at targeting that, both the UK RI Research Council funding in terms of seeding drug discovery awards, and also things like the translational awards that occur at the medical charities such as the British Heart Foundation. And thereafter, the significant scale up usually requires partnership with big pharma, as in the Oxford vaccine program. That's the best example, most recent, of course, example between Oxford and AstraZeneca. George, did you want to add anything from the Oxford perspective?
Well, actually, you said all the important parts, that there is this hurdle that universities are probably best positioned in teaming up with private sector support, startup companies or established companies, to then make that transition from laboratory and basic knowledge to translation and then application and uptake. So I think we all start to understand that this is not a one-man show, but a team sport.
Elliot Byford, perhaps a related question that maybe we might start with you, Shinichi. Elliot Byford has asked, how close do you think we are to seeing this research, that you've all talked about, being translated into successful treatments? Are there any important clinical success stories already that we should be aware of? Can you bring that alive for us a little bit, Shinichi?
Yeah, that's a very important question. We can somehow translate into clinics if we have a very good idea in the research level or in vitro experiment. At least we have a couple of examples in Japan. Usually we use human iPS cells, but the first successful story has been done in the retina. So retinal degeneration has been treated by induced iPS cells, and seems to be clinical trial have been seen to be very successful. So it's a one good example. So, but in order to get such an achievement, several factors would be necessary. First of all, basic research should be done. We need to have a proof of concept by basic research. Sometimes experimental animal models will be feasible. And then we need to do translational medicine. So there we need to have, for example, registry of the patient, especially in the case of rare disease. We need to have a very good registry, otherwise we cannot go ahead. And then application to clinical medicine, mainly in the hospital. So that those combination of basic research and the translational research and the hospital activity, those factors will be really necessary to get real transition from basic research to clinical application. And if we have time, we can raise other cases or other examples by using iPS cells. But in a field like skeletal muscle, as Paul mentioned, still it is not so easy to induce myogenic cells from iPS cells. And then we need to rule out multipotent cells from final products. So a lot of hurdle we need to overcome to translate results of basic research to clinical medicine. That's my short answer.
Thank you. Very good answer. Maybe perhaps to put another question that then also this frames into the future. Martin Hatfield has asked, there's great presentations looking ahead, say 10 years. What areas of public health do you hope will benefit most from the IDRM's work, but from your collaborations? Perhaps that's a good way of looking both at the present and at the forecasting for the future. Paul or George, would you like to?
I can start by just focusing on one area which I will be leading in the institute, and that's in the cardiovascular space. So I think that it's very clear that heart disease is still one of the #1 killers across the world, if you take COVID out of the equation for the moment. But COVID itself and long COVID will be another risk factor for cardiovascular disease through microvascular damage and structural disarray to the heart muscle cells. So it's another factor which will contribute to an increased burden of disease. And this is on the increase in Japan. We're aware of that as well. So I think any attempt to regenerate tissue following a heart attack that could be successful will have a profound effect on public health, because that will prevent the onset of heart failure, which is one of the most debilitating diseases and prominent diseases worldwide, and particularly in the developed sort of Western world as well. So I think, where we're heading with this is to target the initial event, the initial acute injury, which of course is sadly very common, and then lead to regenerative strategies which prevent this remodeling and this progressive disease, which leads to very, very terrible morbidity and, of course, ultimately mortality. And heart transplantation being the only cure is not going to be something that's realistic for all of those people with the disease. That's just simply not possible. So I think this is a potential for a huge impact on a major disease that is now spreading worldwide and is still very prominent both in the UK and in Japan. So that's just one example of a thematic area in the Institute.
So probably, so probably to add on to that, what Paul already mentioned is that the body's initial response to injury in the years after early childhood is typically to form a scar. And this involves the immune system. And so looking forward into the next 10 years, I think one of the goals will be to harness the immune system in such a way that it produces the ability to recapitulate normal tissue development and not just form a scar. And this will mean that we understand how particularly the aging immune system tends to prefer inflammation over repair. And here IDRM will clearly contribute to a better understanding of how we can get this balance right. And I've seen there was one question from the audience asking, so why are then T cells so prone to aging? And is that part of the ticket, or is that something that we have been exposed to because we live longer? And it's most likely the latter. Our overall organ systems had been test-driven and proven to work during a certain period of time, but with our advanced capacity to age, we just start to see limitations of the systems. And for the immune system, this is particularly obvious and is termed inflammaging, namely the immune system tends to create more inflammation as we get older.
Shin'ichi, would you like to add anything about where you think the biggest gain?
Yeah, very few, but personally, as I target aging or muscle wasting, will be a very good target in the next 10 years, as also Paul and George mentioned. But when we consider the application of iPS cells, like cell therapy, spinal cord injury or Parkinson's disease to be a direct target next 10 years. But I should mention one thing. In the world, more than 7,500 of rare disease. So we can somehow overcome like many of or most of rare disease by regenerative medicine. That would be wonderful. And that's my hope and like dream.
Well, thank you. That's a wonderful point on which to bring ourselves to a close. We're fortunately running out of time. I like the thought that that diseases that we didn't need to pay attention to in the past because our bodies prevented us from receiving them because we passed away too soon, now will get more attention thanks to your institute and to your collaboration with Japan. A couple of people, I think correctly, from Great Britain, Sasakawa Foundation, have emphasized the value of researcher interchange between young researchers, and the link for the Butterfield Awards. One of the great ways to encourage that is there in the Q&A, and I encourage you to also go to the Great Britain Sasakawa Foundation website to do that. I think that one consequence of the pandemic has been this loss of personal interchange that we can substitute only in part by Zoom, and but I'm glad that we could very much do so today. And I wish both the Institute for Developmental and Regenerative Medicine and the Neuroscience Institute in Tokyo and your collaboration every success in the future. Congratulations on what you've achieved so far, and thank you very much for sharing your expertise and insight and thoughts with us today. And I hope that many of those who sent very good questions that I wasn't able to get to might be able to send them directly to you to pursue the discussion later. But thank you all, Paul Reilly, Shinichi Takeda, and Georg Holländer for being with us today, and thanks everyone at the Japan Society for attending. Best of luck and thank you. Have a good evening. Have a good day.
Thank you very much.
Thank you very much.